again to another fabulous night of Kaplan's free week of MCAT. Hey, give me a hi or hello. Let me know that you can hear me. And then you're ready to get started with today's lesson. Hi. Hi. Hey, Annette. Hey, Giselle. Hi, Lise. Hi, Kat. Hi, Rain. Hi. Hi. Hey, everybody. You're going so fast now. Hi. Hey. I'm so excited to see everybody. So many familiar faces. I'm glad that you're back for more. We didn't scare you off with some of these tough topics we've been tackling. I'm so excited to see you. Are any of my molecular genetics fans out here super excited for Mulgen tonight? Anybody? Anybody? I'm super excited. This is one of my favorite topics. Not least of all because of the amount of points that are available. I hear you. I hear you. I also love all sciences, but some of them are a little closer to my heart than others. You know, mostly the ones that I'm better at. I am super excited to see everybody. We're going to start, as per always, with a welcome and introduction. Welcome. This is Kaplan's free week of MCAT. Is anybody just tuning in today? You've not been here for the other days of the free week? This is your first day of free weeking with us. I know I see a lot of familiar faces, so I know. And where are my veterans of the week who have been here with us? Well, hi, Cass. Hey, Emily. Hey, Katie. Welcome, welcome. Where are my frequent flyers? Yes. Back for more. Absolutely. Hi. Yeah, baby. Hi. I'm so excited to see all my vets too. Well, listen, if you missed it, I'm sorry. We had a good time without you. We tried not to have a good time without you, but every day is a good day at Kaplan. Here in Kaplan MCAT lands. Well, welcome. I hope that you do feel welcome. My name is Allie. I am a Kaplan Plus coach here. So whenever you look at your Plus courses from Kaplan, they all come with coaching. And sometimes people ask me, like, what is a coach? Like, what is the difference between a coach and a tutor? And, you know, I can tutor and I do help you with topic work if you need that, if that's what helps you get to your goal score in the least amount of time. But my main goal is just that, getting you to your goal score in the least amount of time possible by making sure that we're always using the right mix of resources and study planning and adjusting as necessary. Has anybody had to adjust their study plan already? Like whatever you planned to do originally, whether it be the timeline or the resources or the test date you were aiming for, you ended up having to change it around a little bit. Oh, thank you for being brave enough to admit, Emily. You know what? This happens to everybody, I think, at least once. And that is the thing that I do. That is where coaches come in. We're always going over your test results with you, looking at your homework utilization, looking at how many points you're getting, talking about what's going on in your life so that we can make changes as necessary, figuring out what to study, when, and that's what I do. So that is my introduction. And if you want to work with me or a coach like me, all you have to do is look for those Kaplan Plus courses, because a unique feature of that Plus course is that it comes with a coach. So a lot of good things there too. And also, you know, we have been doing coaching via email as much as we can. You know, coaching with Kaplan is like one-to-one, just like tutoring is. So it's very, very personalized, but we've been trying to do kind of like general coaching via email all week long. Shout out if you have utilized that. Where are my students who've been emailing for advice this week? Because I know that I've talked to a lot of you. We've been talking about study planning and timelining and what resources we have, what should we get, how can we use what we already have, how can we plan for the future, what test dates should we shoot for, this is my current score, this is my goal score, is this a realistic time frame? None of my email coaching students here today? Not any of them? Not any of them? I did send out the email from yesterday, so you should have received it. I sent it out a little earlier. If you did not, there you are. Hey, if you did not receive it, just go ahead and send me an email at consultateacher@kaplan.com. And I will say too that this is the first time that I am disappointed to say we weren't able to get back to every single email before the start of class today. So there are a few that are awaiting response. I'm going to make sure to get back to all of you tonight. So do be on the lookout. And I know y'all aren't shy because I saw some emails coming in at 1:30 a.m. in the morning. So I know y'all are up, stay up late studying. So you will not be surprised or confused to see that I am the same type of person who will also stay up very late to talk MCAT. So watch out for those today. And Becca, listen, feel free, send a long email, send several short emails, any questions that you have, we're happy to advise. And, you know, it's been going really well, I think. And in fact, we've even managed to get some of the students into Kaplan MCAT prep tuition assistance. Or you can think of it as like scholarships because you don't have to pay it back. So one of the things that we have worked on here recently is just knowing the resource options that you have. So we did talk about earlier how in the link right there, kaplanteacherconsult.youcanbookme.com, or by calling 1-800-CAP-TEST, you can figure out what payment arrangements or options are available to you. And one of those fabulous options is that for students who are in undergrad and on financial aid, we use the same FAFSA information that is used for your undergrad financial aid to assess financial need and provide tuition assistance for students needing MCAT prep. And the cool thing about tuition assistance from Kaplan is, unlike tuition assistance or financial aid from school, you don't have to pay it back. It's like a scholarship, need-based scholarship. So if you do fit into that category, if you are an undergrad, if you do show financial need on your FAFSA, maybe you're on financial aid at school and you're looking for MCAT prep and you're looking for a little help, feel free to send us a message and your tutor will talk to you about how to apply for that so we can get that going. But I know that a few of you already, we've had this conversation. Now we can have it more widely. So I hope that's helpful for you. But do make an appointment with the teacher to talk to them, because you have no idea what is available to you without asking. So that's a big part of it. We're going to start with an overview of the MCAT. We always do that because we always have people who are not only new to Kaplan today, but are new to the MCAT. I have never yet asked this question and not had at least one. Who is new to the MCAT today? Who is starting MCAT prep today? This is your first day, just brand new to the whole test. Anybody? Anybody? Are you out there? Of course you are. Always. Hi. So we always start with an overview so that our newbies can put this lesson into context. Now, we're not going to take too long on it because we have really talked about these topics a lot this week because we've been having lessons every single day. But we will talk about kind of a bird's eye view of what the MCAT is before getting into our strategy and content lesson, which today is on molecular genetics. Who did the pre-quiz for today? I think that pre-quiz is so cute. Did you guys like that one? I really like that pre-quiz that we did. That's one of my very favorites. Isn't that neat? I think it's really, the pre-quizzes are really great because they give you the opportunity to kind of do what you need to do on the MCAT, which is take the science that you know and now apply it in a situation where it might not look the way you expect it to look, because it's not a super straightforward question. It's a situation, right? So thinking about situations that are parallel and maybe not just like that rote science. I think it really warms up the brain, and it's a great way to really think about your MCAT, because your MCAT asks you to do the same thing, a little bit more difficult than the pre-quiz that I sent you. But I hope that you enjoyed it, because that is one of my favorites. So today's lesson is taught by your favorite teacher and mine. Teacher Petros is back again for molecular genetics. It's got a lot of really great things to help you with. So we did, we have transcription, we have translation. We did one in the pre-quiz. We'll be doing the other in the lesson tonight. So we're going to get to hit a little bit of both. And so you have a little bit of extra notes on both, a lot of extra notes from today's lesson. I also sent last night the notes on memory. So you should have gotten those notes on memory. I hope that you find those to be helpful. I tell you what, it's such an important concept, but I think that we kind of covered it in the notes that I sent. So make sure to memorize that and get into some MCAT questions about memory and start practicing what you know in that MCAT application form. Coaching tonight is going to be best practices, the do's and don'ts of test prep, because tonight, I know, try not to cry. Don't cry for me, Argentina. Tonight is my last night with you on the free week of MCAT. And so I want to make sure that I don't leave you without making sure that everybody gets my overall best practices, do's and don'ts in terms of making your study plan. I want to make sure that even when I'm not with you, you have those bullet points that you can hold up against what you're planning for yourself, and just make sure. And we'll talk about all of the whys, and we'll, oh, you guys are so sweet. We'll talk about all of the whys. And also, if we have time, I want to do something really, really cool. I'm going to miss you guys too. I really will. But you can always email me. Like, I'm never going to, even though it's not the free week, I'm not going to turn off and just be like, no, never. But, what was I going to say? If I have time tonight, I do want to show you guys too, inside of the Kaplan course resources. So I know that you guys have heard a lot about, we have X and we have Y and we have Z, and you can see it on our website. But I think sometimes, do you guys find that it's like overwhelming, the amount of resources? It's like, it's so many, and to try and read about it, it seems very like, how does that fit in? Is that too much? How would I utilize that? Like, how do I see this? Like, just conceptually, I think it can be a little overwhelming. So I want to show you so you can get that visual, so that you can, see what it's like. And so I've been, in a lot of the emails that we've been sending back and forth, I've been sending pictures of kind of the Kaplan resources and describing how they work. But tonight, I want to take y'all live inside of the Kaplan class, where we can discuss all of the features, and you can see it in person. So if there's any resource, be it the adaptive QBank that we've been talking about, or you guys tell me, what resources have you guys been researching? I know a lot of you have been researching the adaptive QBank, so that is something that I will take you inside of tonight. A lot of students looking at the difference between plus and non-plus. We talked about that last night too, so that's something that I'm going to be able to show you the difference on the MCAT channel. We'll be seeing that too in action. It won't be live anymore because MCAT channel ends tonight at 10 and we end at 10:30. So we won't get to see them live, but I'll show you their archive, our practice test, how we integrate the AAMC materials, all of that. So I'm very excited to do that as my last thing with you. And then I will leave you in the very capable hands of Teacher Petros to go on for my two favorite MCAT lessons, which are the boot camp, which you have to be signed up for. Please tell me right now you're signed up for the boot camp. It is fabulous. You got to do the boot camp. You're going to go over some of the hardest, most commonly missed topics and concepts on the MCAT. But we're going to show you how to apply that test-taking strategy with the content that you know. So we're going to show you how to cut through the convoluted questions to see the simple science underneath, so that then you can apply that content that you have memorized. Okay, so we're going to be doing that. And then if you have not yet taken a practice test, is there anybody here who has not yet taken the Kaplan practice test? If you have not yet taken the Kaplan practice test, shout out, let us know. Out yourselves. I'm just kidding. We love you. We love you. But if you have not yet taken the free practice test from Kaplan, that is happening Saturday. So make sure that you sign up for that, because the lesson after the practice test is honestly my favorite free lesson. Of all of the free things that we do, that is my very favorite free lesson, because it really shows you, you get the opportunity to take the test. So you've thought your way through these problems. You've seen how you approach and how long it takes you to do that approach and how you thought through it. And then the lesson now covers those questions so that you can see how we will do it and the approach that we will take. And you'll get to see that combination that we keep talking about, is how do you combine test taking strategy, MCAT science skills, which are very different than just science skills regularly, with the science content that you know. And so just do not miss it. And of course, if your real test is Saturday, then you already know how to do everything that you need to know, and you are going to do fabulously. That's for you, Ms. Harley. I have every confidence. Let's get into it. So the MCAT is obviously very important. You all knew this, right? Like, I don't need to spend too much time telling you that the MCAT is a really important piece of your application packet. I think that you know that. It is the most important factor in your admissions process. Number 2 is your undergraduate GPA. So you want to really look at the combination of both, but the really good thing is that the MCAT score is a higher percentage. Did anybody ever get a bad grade? Anybody? I know some of you are like, no, but I am brave enough to admit that I am not in the no category. I definitely did not always get perfect grades. I tried my best. Sometimes it didn't work out for me. So that was the thing, right? Is there anybody who has not been able to get in as much volunteerism as they wanted to prior to applying? Because that was also me. I'll tell you, when I was preparing for my entrance exams, I was a single mom, and I was going to school full time. I was working a lot of different things. So I didn't get to do every single thing as much as I wanted to. Is there anybody who, let's see, what else can we do? Anybody who hasn't got to have real life experience as much as they wanted to? Like maybe you didn't get to shadow enough. Maybe you didn't get to step in there and get your hands dirty on some stuff. Oh yeah. Yeah, and what about research? Is there anybody who doesn't have a lot of research to show? Because these are all things that happen. Or what about personal or letters of recommendation? Has anybody, I know a lot of us are in school, most of us are in school, but are there any of us who have already graduated and maybe don't have that same relationship with our professors that we could get those we can get those letters of recommendation anymore from our professors, because they do prefer professors. So we would want to get them from professors ideally, but we're not all able to do that. See? So we all have a little bit of something that we would like to make up for. And that's why I love this test. And I love how much importance they put on this test. It is very easy to look at the MCAT and say, it's so unfair, it's worth so much, I'm so scared about it, and get very negative about it. But the MCAT is what I like to think of it as the great equalizer. It is the thing that is, the thing you can manipulate the most. Because even if you're still an undergrad, you're not making a big change to your GPA between now and whenever you finish this semester. Your GPA is pretty much set. One semester anyway isn't going to be a big deal. It just is what it is at this point. But your MCAT, you can go from a low percentile to a high percentile if you put the work in. That can happen in a short amount of time. Your MCAT is the thing where you can say, you know what, I know that person has better grades, but look, I'm just as good. I know that person has stellar letters of rec, but look, I'm just as good. I know that person has tons of research, but look at me. Look at my MCAT score. And so that's why I actually really, really love it. And I really like to always look at it in a very positive light and really look at it as that opportunity. It is a hand reaching out for you, because they would love the person with the most research and the bestest grades and all of the volunteerism and all of that, but they will also take the person who works hard, kicks butt, takes name, and provides that MCAT score that says, you know what, I'm here to work, I'm smart, and I am meant to be a doctor. And I think it's great that they give us that opportunity, because we don't all get the opportunity to do all of the other pieces completely or as much as we would want to. And I would think that it would be very unfair for them to say, oh, well, too bad. Too bad you have a life that means that you don't get to do all of these different things, and too bad you didn't have time for that. Oh, well, you're not meant to be a doctor, because that's not true. That's not true at all. And so I really, really like to look at this test in a positive way. So I hope that encourages you. And whenever you do feel down about the MCAT and it starts to stress you out, remind yourself of the great opportunity that you have, not only to get into school, but also to not only to get into school, but also to get financial aid from school and to get into multiple schools and really have that opportunity to shine and stand out in the crowd. How do you become a good standardized test taker? Yes, absolutely you can. The thing is about it, it's standardized, right? Where are my psychology people? What is wrong with a standardized IQ test? This is not an IQ test, but let's talk about IQ tests for a minute. Psych folks, those of y'all studying psych, tell me what's wrong with a standardized IQ test? What is wrong with it? Hey, sis Cass, are you one of my psych folks? Are you one of my psych folks? All of those things, absolutely. Your IQ is something that is what it is, right? When you take a standardized IQ test, it's not. You either have had some experience with some of these things which make you do better, right? There you go. It's not equitable. Or if you have not had that experience, and yes, absolutely, it does not measure true intelligence. If you have not had that experience and you go and you take the standardized, like the Mensa test, right? So you go and take one of Mensa's IQ tests, standardized tests somewhere in this country to get into Mensa, right? You don't get quite where you wanted to do to get to Mensa, but then you go and take the same test again in a month. Are you going to do better on that test? Absolutely, because it's standardized. The fact that you have standardized it means you had to leave mile markers. You had to leave same patterns. You had to leave same clue words. You had to do things in order to prove that one person's score from June of 2011, it means the same as that same numerical score in June of 2018. And the only way that they can say that is by proving that they left enough patterns, they left enough markers in the same kind. So that's right there, that's where you come in with standardized testing. And this is all standardized testing, anywhere, anytime, but especially the MCAT, is that they have been giving this test for a very long time, and they have told every medical school in this country that their test is valid and that they can look at, you know, my 514 score and they can compare it to any other 514 score, and they can say those two people are comparable, even though they had a test with totally different questions on it. And the only way they can do that is by saying, yep, we gave her just as many clues as we gave him. We gave her just as many patterns as we gave him. And that is where test taking skills come into play. And I keep telling y'all over and over and over again, you have to take your content knowledge and apply it on MCAT questions. And that's why you have to see the common wrong answer types that they use. That's one of the ways they say my score is the same as his score, because they say, hey, well, we put the same wrong answer traps on both of them and she fell for it and he didn't. Those are the things, right? So practice your MCAT format questions, learn to understand the test and how it works, and you'll be able to beat it and you'll be able to increase your skills, absolutely, because standardizing means they have built-in patterns. Standardizing means that they have made the tests same in a certain way, even while using different questions. So it is that sameness that they have built into this that you can take advantage of, huge advantage of. I am much better at using those patterns and strategies. I'm actually a lot better at that than I am at content, and I always have been. but it's okay because they also like that person, the person who thinks strategically, who sees patterns, who prioritizes their work accordingly. And that's where you can come in. So yes, absolutely, you can do better on standardized tests, and you should put the time and the practice into doing that. The reason a lot of us don't have test taking skills is because we have not really been taught them. I talked about this earlier in the week too. Have you guys ever heard, when they're doing standardized testing in the year for like undergrad, for like high school, and they'll say, that teacher only teaches for the test. That's all they do. They just teach for the test. Have you ever heard that? Have you ever heard that? They just teach to the test. They just teach to the test. That's all they do. And it's like a huge insult. It's a huge insult to those teachers. And so teachers hate to teach to the test. They hate it. And what that means is that you have not been taught test-taking skills because they don't want to be teaching to the test. But guess what? You can teach to the test. And I do teach to the test. And I take that as the highest compliment because I teach to the test, and that is what we do. We are test prep experts. My job is not to make sure that you're ready for medical school. I hope you do great when you get there. I'm sure you will. I'm going to trust that your undergrad professors prepared you for that. My one and only job is to teach for the test. So absolutely, I would be way out of business. I would be way out of a job if you could not get better at standardized tests. So great question, and very enthusiastic answer because that is one of my favorite topics. And yeah, you can fix it. And the reason that you might not be good at standardized tests is not anything to do with you or any kind of deficiency. It's because they just don't teach it. They just don't teach it coming up. They consider it a huge insult coming up in school and education, but we don't here. We don't think that that's an insult. We think that's a really big compliment, actually. We teach to the test all the time, every day. What is tested on the MCAT? These are your test sections, questions and links. Make sure to take a screenshot. Make sure to take a screenshot of this. But tell me this, test sections. Does that mean that those sciences are only going to be held within those sections? That you're not going to see any chemistry coming into psychos, you're not going to see any crossover ever between the different sections and their titles? Those of you who've been studying my experts, let me know, shout out. Heck no. Oh, no. The MCAT loves to put some MCAT in your MCAT. So you can expect that the sciences are going to cross over. That's another reason to be practicing MCAT style questions as much as possible so that as you're reading about your chemistry, you go do some chemistry questions and you see the other sciences they're bringing in. You see how they're bringing it in. And then you can learn to mix the two as well. Because if you only study your content very discreetly, then what's going to happen when you're taking a timed, scored practice test or real test, God forbid, you're going to see where they've mixed it and you're not going to be prepared because you're so used to seeing them discreetly. So make sure, again, it's that MCAT style targeted practice that you've got to get done. All right, let's talk about how it's scored. 500, tell me, what's that score? What's that magic score for MD programs? That average admit score. What is it? What is it? Nicole, very good. Very good. Yes. There you go. That range, 509 to 511. So we like to call it a 510, but it is a range, 509 to 511. That is your average admit range. So you'd be safe to call it a 510. That's for your MD programs. That's not to say that that is the target score for every program. You have got to do your research into your programs that you want to go to, find out what their average admit score was, and utilize that information to really target what you're doing. I actually use, I don't know if any of you guys have purchased this, but I actually use US News and World Reports. I don't know how much it is, and I don't know if like for a single person, if it's worthwhile. I think it might be like 20 bucks, but I have a subscription there and I can see all of the rankings for medical school, but not only that, I can see all of their average admit scores, all of their diversity statistics, how many women, how many men, what kind of backgrounds, how much research. I can see Gosh, what else can you see? Emails, phone numbers, deadlines, like anything about the school, what type of research they're doing, anything you want to know, you can find out all in one place about all the schools at US News. So that's what I use. But do your research. If you know, like already know the programs you want, and you don't want to see an overview of all the programs, then make sure and just do your research. Google is your good friend, and figure out what it is that they are looking for. That is usnews.com. US News is the ranking body for education. It is the gold standard in ranking for schools in a lot of people's minds. So they keep a lot of data, and you can have access to all of that. So I think it's like super helpful, but it's a really good way. You can also just look at the list and the rankings and just use that, and then Google and go find the school. But it's all about the research now. Figure out what they're looking for, and then make yourself fit that mold. Here you go with your pretty picture. I told you they love to put science in the science. This rainbow chart will show you that. Go ahead and take a screenshot here. This gives you that bird's eye view of how they're putting that science in the science. All right, y'all. It's time. I know you're going to miss me. I'll be back for best practices, do's and don'ts of study planning, trying to make sure that you have the things to avoid and the things to employ in order to make sure that you get your score in your time frame. And it will be my last coaching session with you for the week. So we'll make it a great one. We'll make it a valuable one, and we'll answer a lot of questions about what you are going through and what you are planning. So I look forward to it. But right now, it's time for Teacher Petrus's lesson on molecular genetics. I'm sure you're going to love it. Enjoy. Bye-bye.
All right, everyone. Welcome. Hopefully you're all doing really well. Getting the camera to turn itself on. That happens sometimes when we switch over cameras. How's it going? So thank you, Ali, for the intro. Good stuff. I will say this much: the MCAT, probably the most important thing that you're going to need to worry about right now as far as your preparation is concerned. As I mentioned, my name is Petros. It's good to meet you all. Hello. I am a teacher here at Kaplan, have been so for now 19 years. I'm also the director of our pre-health program, so I actually oversee all of our MCAT courses and kind of all the work that we do in creating our materials and creating our courses and creating all the study plans and all the different programs that we have. So if you got questions about anything MCAT related, we're all here to answer them for you. So Shwarya is here, Allison's here, Candace is here, Mackenzie is here, Sharon is here, all teachers here at Kaplan who I've had the pleasure of working with at least for a number of years, and you all have the pleasure of working with for the duration of this evening and this week. So with that said, we're going to dive into molecular genetics. This is a very, very specific lesson. This is a lesson that comes from the MCAT channel. The MCAT channel is part of our MCAT program. The way that our MCAT program is designed is we've got a kind of core set of lessons that everybody goes to. And then we've got the MCAT channel, which is 90 individual 1-hour lessons. They're run, all throughout the week. So, like right now, I actually just taught an MCAT channel session and then came over to see all you guys. So this is a series of classes that are taught kind of on rotation throughout the year. The reason why we do it that way is that you can pick and choose. There's, you saw the test is a long test. It's got lots of different topics on it. So you can pick and choose the sessions that are right for you, because really your MCAT preparation, I think the one kind of top bit of advice that I'll give you right from the get-go is that you really want to make sure that it's personalized, that you do the work that you need to do, right? It's about what your individual needs are. I think it's perfectly normal, and I do this myself, where we say, oh, my friend is doing this, I'm going to do that too because it seems to be working for my friend, so I'm going to do that, but maybe that's not the right choice necessarily. So really important to make sure that you're kind of thinking about the personalization. And the nice thing about the MCAT channel is you actually get access to our top instructors from throughout the country because it is delivered live and online. There's a very small group of us that teach on the MCAT channel and work on the MCAT channel. I think it's about 20, 25 people all together. So, you get a good opportunity to really interact with folks. I saw a question coming in from Abrizi. I'm sure that's not your full first name, whether or not you can purchase the MCAT channel separately. Unfortunately, not right now. So it is only available as part of the overall program because it's so integrated. It's so integrated into the rest of the lessons and the rest of the material. And if you got questions about those things, let us know. We'll be more than happy to answer those for you or set you up to talk to somebody who's the right person to talk to about that and figure things out. But the topic at hand is molecular genetics, and specifically we're going to look at transcription and DNA. So we're going to look at kind of the first part of molecular genetics. It's actually a series of sessions that we have related to molecular genetics. We're going to do one of those sessions here tonight as part of free prep week. So here's what we're going to look at. We're going to take a look at the events that are involved in DNA replication. So really like what's going on. We'll talk a little bit about the mechanisms of DNA repair. And I'm just going to put this out there. There are tons of details involved in DNA repair mechanisms. And if you've taken an upper division or an advanced level biochemistry class, you've learned all of those details. That level of detail is not tested on the MCAT. So hopefully that is a relief for some of you. I know it was definitely a relief for a lot of students when we say this to our students who say, "Oh, thank goodness, I don't have to worry about all those little details." So we're going to talk about those mechanisms and just really what you need to know for the MCAT, and then we're going to spend most of our time here talking about transcription and gene regulation because that's really the big topic when it comes down to DNA. So let's look at the DNA, our good friend DNA, and we're going to look at what we call the replication fork. So this is the mechanism for replication, and as hopefully you've kind of already figured out, we like to be very, very interactive here. So we want to hear all your questions and also all your answers to all the questions that we're going to ask you throughout this process. So when we're looking at DNA, the first thing, of course, we remember is DNA is a double helix. It rotates on itself, it folds on itself. But when you want to actually replicate DNA, you got to open up the DNA. That's the bottom line, is you got to make sure that you're opening up the DNA. So we are focusing on a very, very specific part of a strand of DNA. It's called the replication fork. This is part of a larger replication system that's actually happening, and we'll take a look at this as we go. So you got a lot of enzymes that need to take a role in the process of DNA replication. So first, of course, is our good friend helicase. What does helicase do? What is the purpose of life for our little friend called helicase? Why does helicase in fact exist? Why did helicase show up, and why did helicase wake up in the morning? It's there to unzip, right? So helicase's job, its primary function in life, is to unzip the DNA. Now, if you think about the DNA as being twisted on itself, kind of like two strings, if you were to start to, you got it fixed at the bottom, and you start to pull apart those two strings, and you're opening up, you're unzipping your helicase, you're unzipping those strings that are twisted on each other, what's going to start happening to those strings? Or think of your shoelaces, right? If you were to start to pull on the top of those shoelaces and it just kept pulling on the top, what's going to happen to the twisting that's down below? What's going to happen? It's going to do something. They're not going to come back together per se, but that twist is going to get tighter and tighter, right? It's like, don't do this, right? But if you were to twist your hair and you keep twisting your hair, it's going to become a tighter and tighter knot, right? It's going to be a lot of tension that's going to come into play there. So helicase does a great job of unzipping the DNA, but it does an even better job of creating a whole lot of tension. So topoisomerase comes into play to relax the DNA. So if you think about, if you were to personify topoisomerase, it is the relaxation expert, right? It's the person, it is the enzyme that speaks with a calmer voice, right? Maybe has some soothing music on, and what it's doing essentially is it's causing the DNA to relax so that you don't get that kind of super tension, that super twisting that can ultimately come into play. So helicase plays an important role, topoisomerase plays an important role. All of these enzymes play a very important role, and they all really have to work together. If you just unzipped, but you didn't have anything else, then it's worthless, right? If you just have DNA polymerase, which we'll look at here in a second, you didn't have anything else, it would be worthless. So all these things are going to work together. Now, primase comes into play. As its name implies, what do you think primase is going to do? What do you think is, what is primase? What is primase doing? Love the corny jokes, Ashwarya. They're wonderful, wonderful. They're going to add a primer, right? So a primase is going to come in and say, okay, let's start priming you, right? We got to get you ready. So the helicase unzipped the DNA. Topoisomerase has relaxed the DNA. Now the primase comes in and says, I'm going to get you ready for replication because we're going to have to, get down to business here in a second. And so the primase comes in and does that work. And then DNA polymerase starts doing its thing. And DNA polymerase, as the name implies, is going to actually create, it's going to bring in new nucleic acids and create new DNA. Now, the thing about DNA polymerase, and this is important because this will show up on the MCAT, and they will try to trap you with this little tiny detail about DNA polymerase. DNA polymerase, as awesome as an enzyme as it is, it's actually kind of, it's one, I wouldn't say one-sided, I would say it's kind of stupid in that it can only, and write this down, don't forget this, it can only read DNA in the direction of 3' to 5'. It can only build the DNA from 5' to 3'. So what the DNA polymerase will do is it will start building new DNA in the 5' to 3' direction. So it's going to be going over here, it's going to be going like this, right over here, it's going to be going like this, and it's only going in one direction in this process. Now what happens though in this replication fork is that since the DNA polymerase can only be building the DNA in one singular direction, that means that over here in our picture on the left side, this is a continuous creation. This DNA polymerase is going to just keep building. It's going to be grabbing nucleotides, grabbing nucleosides, grabbing nucleosides, grabbing nucleosides, grabbing nucleosides, and making new DNA. Over here, it's going to make a little bit this way, and as the replication fork opens more and more and more, it's going to have to jump back and then make another little piece. And then as it opens up more, it's going to jump back and make another little piece. So it's going to have to keep jumping back. Now, granted, there are multiple DNA polymerases that are working simultaneously because this would take forever if you only had one DNA polymerase in any given scenario. But the moral of the story here is you create all these little tiny pieces that then need to be stuck together. What are these little tiny pieces called? This is the bonus question of the evening so far. What are these bonus questions called? What do they do? They're called Okazaki fragments. Very good. So nicely virtual high five. They're called Okazaki fragments. So named after the individual who identified them. So what's happening is these little tiny fragments, these Okazaki fragments, get stuck together, and ligase is a glue. So the polymerase, it's the muscle. The ligase, it's the glue. It's going to stick it all together, and it's going to bring it all together. You've also got RNA primers, and the reason that you have the RNA primer really comes down to very, very simple mechanics. You are creating a new piece of DNA. So what are you going to prime things with? You're going to prime it with this RNA primer. But also, as you think about this, and as you're kind of looking at the replication fork, part of all of this really comes down to the fact that you have a bunch of things that are happening all at the same time, right? They're happening all at the same time. And so the primer, it's a short little strand of RNA. What it's doing is it's a starting point. It's saying, here's where we're going to begin. So it's required because the enzymes that are going to get things going, they can only add to an existing piece of nucleic acid. And so the RNA primer, it's like the seed, right? It's the catalyst of getting all these things going. So think of it like, think of it as the initial building block, if you will. So you got the RNA primer that serves that role. And then you've got some single-strand binding proteins. The single-strand binding proteins, I wouldn't worry about them too much as far as the MCAT itself is concerned. But essentially, what you're doing there is it's, as the name implies, the single-strand binding proteins are binding the single strands, right? They're kind of keeping things in order, keeping things in check. Okay. Now, when we look at the replication fork, you have these Okazaki fragments on this side that have to get stuck together, but you have a nice one solid piece over here on the left side. So we call this the leading strand, and we call this side over here the lagging strand. And the reason why it's the leading strand versus lagging strand is that the lagging strand lags because that polymerase has to keep jumping back and jumping back. So generally speaking, the way that it's categorized, and this is just somebody at some point decided that this would be the convention and it became the convention, the direction of replication is considered to be the direction that the leading strand is going in. So that's why you see this over here. It says the direction of the replication fork is going upwards like this because the leading strand is going upwards like that as well. All right. So that's the bottom line with DNA and the simple part of the replication fork. Now, you look at eukaryotes. Do eukaryotes have a relatively high amount or low amount of genetic information? What do you think? High or low? What do you think? What do you think? It's pretty high, right? We got a ton. I mean, humans, gosh, we've got 23 pairs of chromosomes, right? We got a lot of genetic information. So if you wanted to replicate your DNA and you just had one replication fork, it would take ages. So here is an adaptation that eukaryotic DNA has, and that is you have multiple origins of replication. So there are multiple ORIs. I like to call them MORIs because there's more of them. Just one way to remember it, not necessarily the best way. It's how it works in my head. Maybe it'll work in your head. But the idea here is that you've got these multiple little replication bubbles. Each of these little Y shapes here, here, these are those replication forks. Notice, though, that because of the nature of DNA, the 5' to 3' direction, if you're leading over here, that means you're leading over here, right? Because you're always, the way that DNA polymerase, of course, works is that it can only build in one direction along the way. Now, at the same time, when we look at our good friend DNA and we look at the nucleotide sequence of DNA, you got chromosomes. And chromosomes, of course, are the condensed, organized version of your chromatin, right? So chromatin is how your DNA is stored in the interphase, and during replication, you've got chromosomes that the DNA condenses into chromosomes. The problem that you run into when you're condensed is that these tips of these chromosomes are extremely vulnerable to damage. So not to equate the chromosome to a car, but what do you have on your car that protects you from little taps that you might get in the parking lot, or it doesn't, it gets scratched a little bit, but at least it's an easily replaceable part. What does your car have? It's got bumpers. Exactly, Emily, right? It's got bumpers. So the telomeres, and again, I'm not trivializing this. This is just a nice way to remember it. Telomeres are the bumpers for your chromosomes, right? You got the bumpers on your car. They protect you from the main part of the body, the main part of the car body being damaged. DNA's got telomeres, right? That's what it is. So the chromosomes have got the telomeres. So this is a nice bit, right? So this is a nice little kind of addition, if you will, that we have, a little protection system so that we don't lose any genetic information. We've got these RNA primers that are over here, but if they get lost, they get lost. Who cares, right? Who cares? We'll replace them with something else, but at least we don't lose the genetic information. So this is actually part of the reason why we've been able to retain our genetic information for as long as we've been able to retain our genetic information. Now, when we look at DNA replication, it's a pretty, it's a really nifty thing. And of course, there are courses that last full terms that talk about DNA replication. So the fact that we're going to try to do this in, an hour is definitely not. There's so much more. But as far as the MCAT, that's what you need to know. When you look at DNA replication, it's referred to as being a semi-conservative process. And the reason for this is because in every new piece of DNA, you always have one strand that is old, and you have one strand that has newly been created. Okay, so you always have an even mix of the two. And there were a series of experiments numbers of years ago, far before any of us were born, which were trying to theorize which one it was. Messel's and Stahl experiments is what they were referred to as. They had proposed that there might be conservative, it might be semi-conservative, which is what they ended up finding that it was. Conservative replication would imply that all the old sticks together and only a new piece is made, which we saw earlier why that's not the case. And then dispersive is where it kind of gets mixed up. So don't worry about these two because they have never actually been seen, or they haven't been seen as being biologically significant, meaning we can't really find good examples of them. Semi-conservative is what we have, right? It's a semi-conservative replication as far as the DNA is concerned. So we've talked about DNA. We've talked about the order of how things work, right? We have to unwind, we have to unzip, we have to do all the things in order to get things to happen. So take a minute, we'll put a minute on the clock, and I want you to answer this question in the poll, which is a simple one. What's the order that DNA synthesis is going to ultimately follow? Good luck. All right, so this one was a good warm-up question for sure. So I'm glad we did it. It looks like 100% of you, now 98% of you, have chosen answer choice C, and you'd be absolutely correct, right? The order is you unwind, right? The DNA gets unwound, and then you've got the pairing of the actual nucleotides, and then it's joined back again, right? It all comes back together and does its thing. Good warm-up question for sure. Let's try this one. I'll give you guys 45 seconds on this one because I think you're not going to need as much time on it. So try this one out. So this is a little bit tougher. What's not required for DNA replication? Well, we've got a lot of stuff that's going on in all of this. Actually, all of these are required for DNA replication. So let me walk you through this, what we mean by this. So deoxynucleoside triphosphates, those are the pieces that are being grabbed by the DNA polymerase to actually come in and become one of the base pairs, right? So deoxynucleosides, super important. You've got GTP, right? Part of one of those. You have guanine, cytosine, adenine, right? All of them. That's what the DNTPs are. ATP, definitely necessary. This is a high energy consumption scenario. And then you definitely need a primer, right? You need the primer. Remember, the primer is there because the only way that the DNA polymerase can do its thing is if it's attaching itself to something that is already a nucleic acid. So it needs the primer. So none of these are actually are going to not be a requirement. So here's the thing about the MCAT. This is a small question, but it's actually a big question because they're not asking you which one is a requirement. They're asking you which one is not a requirement. And in fact, the three that they list out are all requirements. Therefore, none of the above is going to be the correct answer choice. I see about 57% of you or so chose B, definitely very power driven. Only 27% of you chose answer choice D, which is the correct answer. None of the above, because you need all three of these pieces in order to make this happen. So here's the deal: when you look at the MCAT and you think about these things, the wording sometimes does get you. And Ben, I'm with you. Answer choice D says none of the above, as if none of the above are needed. No, none of the above are not a requirement, right? For DNA replication, and the MCAT loves to use this as an answer choice. So Emily, I see you're asking, do they ask a lot of these? Yeah, they totally do. It's a relatively easier way to write a question, but it also tests you in a better way because in fact they're testing you on much broader general knowledge in making that happen. All right. So that's what we got there. Very nicely done. Good try, but now you know where to go as far as the next one. So this learning goal is a little smaller. Not gonna lie. It's one slide. Bottom line is that DNA replication is not perfect, and DNA does get damaged, right? So there's damage that can happen to DNA, and the biggest cause of damage to DNA is UV rays. So, if you were here last night for Light and Optics, out of curiosity, was anyone here last night for Light and Optics? I feel like there were not too many of you earlier on when Ali was asking if you were here before. We had a lot of new people, but maybe there were some of you that were here for Light and Optics. We talked about UV rays. Why are UV rays bad? Well, they're bad because they damage your DNA, right? That's why they cause all the problems that they can cause. Good, there were a bunch of you that were here. I was gonna say your names looked all familiar, and in fact, you got your pictures here, so I recognize you. So the thing about DNA is that yes, it can get damaged, but it can also repair itself. The thing about DNA repair is that it's a very, very crude system when you think about it. And here's what I mean by this. So here's a scenario where this UV ray has caused damage to these two base pairs, in that instead of the T pairing with the A, the T's decide to pair with each other, leaving the A's all alone. Rather than having a mechanism that is specific to cutting this bond apart and then reattaching it back to what the T is supposed to be attached to, instead, what happens is the DNA repair system, this exonuclease, so excision endonuclease is an exonuclease, what it does is it just cuts out the whole part that's bad, right? It's kind of like if you have a nick in your wall, and instead of just patching up that little piece, you just take out the whole piece of the wall and put a whole new piece in. That's what this is doing. It's saying, okay, I'm just gonna take out kind of a whole chunk, and what it does is a couple base pairs to the left, a couple base pairs to the right. It cuts out this piece, in comes DNA polymerase, and we know that DNA polymerase can build DNA all day long. And so DNA polymerase builds a nice new little piece here, and then you've got this nick, and who's going to glue these two pieces back together? Who's the gluer in our process? Who's the gluer in all of this? Who is making this happen? It's ligase, right? It's ligase that's going to make it happen. So bottom line, we got the ligase that's coming in, and it's going to glue this all together, and that's what we have, the repair that happens. So, you've got UV light, X-rays can also damage DNA. You've got the removal of the damaged DNA, and then you just close it back up with polymerase and ligase all over again, right? That's the bottom line. So here, we'll do this one together here. All of the following DNA replication enzymes can also be utilized in DNA excision repair, except for which one? So this is one of those, again, another funky one, right? So instead of the being, which one is not, it's one saying except. So all of the following DNA replication enzymes can also be utilized in DNA excision repair, except for which one. So go and put your answers into the poll. We won't even put a timer for this one. So we've got endonuclease, DNA ligase, and DNA polymerase. So go ahead and put your answers in the poll. Let's see how this one plays out for us. Let's see how this plays out. All right, so looks like 65% of you are choosing answer choice D, and you would be absolutely correct in choosing answer choice D. All of them are necessary for DNA excision repair. You need endonuclease, you need the ligase, and you need the polymerase. So these are all enzymes that are involved in DNA replication, and they are involved in the excision repair. Here's the cool part of this. This makes it much easier for us in the long run, right? It makes this easier for us because you think about it and you say, okay, bottom line is you don't need a separate set of enzymes to make that happen. Now, a couple of you are probably wondering, I haven't seen any questions about this yet, but I'm guessing this is in your mind. You're like, wait a minute, Petros, you didn't show us endonuclease when we were looking at replication. Well, guess what? When you are replicating DNA, when you're creating new DNA, there is a proofreading step that also happens. And during that proofreading step, there is repair in the replication itself. So in fact, both processes use the same exact set of enzymes, which is good for us because that means your body doesn't have to create, or there isn't a whole other set of enzymes that would be created. And it's really good for us because that means you don't have to memorize another different set of enzymes as far as the MCAT is concerned. So you can thank the body, right? You can thank the body to thank the body to make it one less thing that you have to worry about memorizing. All right. So endonuclease definitely shows up in DNA replication. It's in the proofreading. It's in the proofreading step that's there. So similar to like when you write a paper and you proofread it. You're the endonuclease when you're proofreading yourself along the way. All right, cool stuff. So that's the second learning goal. As I said, it was small. Now we're going to shift to the big one, and that is the central dogma and this whole notion of how and why we are doing all the things that we're doing. So DNA, those are, that's your genetic information. But DNA needs to somehow be expressed. And the way the DNA is going to be expressed is, of course, through proteins. But from DNA, you can't code for a protein. So you need somebody in between. Who's the somebody in between? Who's the somebody in between that's going to come and save the day so that you can get from your genetic information to the proteins that you ultimately need? Good old messenger RNA, right? Good old messenger RNA. That's the big one. So that's what we're going to look at here. We're going to look at this portion of kind of the circle of life, which is good old transcription. All right. Now, before I go any further, I want to clear one thing up and clear this up so that you don't make this mistake on the test. The MCAT loves to confuse test takers like yourself by interchanging the words transcription and translation in answer choices. So here's a nice, simple way that you can write this down. Hopefully this will help you. You maybe have heard it before, but transcription is the act of taking something that has been said in one form and transcribing it into another form. So, for instance, if I were to say to you, transcribe everything that I'm saying, you would write down every single word that is coming out of my mouth, and you would do so in English because I'm speaking English, right? So I say 1, 2, 3, 4, 5, you would write down 1, 2, 3, 4, 5. You would be transcribing what I'm saying. If I then said to you, now translate this, you would then take that and put it into a different language, right? You'd say, okay, I'm going to translate 1, 2, 3, 4, 5 into Spanish or into French or whatever language, right? You would take the words that I said, have them written down in English, and then translate them into a new language. That's the exact same thing that's happening here. DNA gets transcribed to RNA. DNA is written in the language of nucleic acids. RNA is in the language of nucleic acid. RNA then gets translated to proteins. The RNA doesn't become the protein, right? The DNA doesn't become the RNA. The RNA gets translated to proteins, and you're going from a nucleic acid to an amino acid. You're going to a new language. So don't let transcription and translation confuse you, because that is a commonplace that that happens on the MCAT. They love to trip you up. Do you like that analogy? Because if you don't, I'll stop using it. But I've been using that for a while, so we're gonna keep using it unless you tell me not to. Just remember your language lessons, right? Remember your language lessons. That's what it is. Is that good? Okay, good. So bottom line is that you've got to make sure that you're keeping things in order, because they will try to mess you up, right? They will try to mess you up along the way. So let's take a look at transcription, and specifically, we're going to take a look at even gene regulation along the way. So we're going to use a prokaryote. We're going to use the lac operon to talk about gene regulation. The bottom line is there are going to be times where you need a gene to be expressed, and there are going to be times where you don't want the gene to be expressed, because maybe too much of the gene being expressed is actually going to cause toxicity. So in the lac operon example, what you have is you have a region that is your operator, and this region is going to have a repressor on it. And if you don't have some kind of inducer, which in this case specifically is lactose, the gene is going to be repressed, and the lactase is not going to be transcribed. So think about this from the perspective of the reason why you would want to transcribe the genes for lactase is if you've got lactose around, then you need the lactose or the lactase to break down the lactose. So if you don't have any lactose around, there's no point in expending all this energy, all this work, right, in order to make the lactase. So you don't make any. But if you have an inducer, so you have the lactose present, then what will happen is the inducer will in fact bind itself to the repressor, effectively repressing the repressor and opening up this region over here, which now allows the RNA polymerase to do its thing. It can do the transcription, and it can do its thing, and it can start making the messenger RNA, which will then go and be translated into the protein, right? So that's what we have as far as these guys are concerned. So with the inducer, the gene can now ultimately be transcribed. At the same time, in the trip operon, it's similar story but a little bit different. The repressor in this case, and these are just two different models for gene regulation. In this case, the repressor can't bind to the operator by itself. And so what happens is, without the repressor, the gene is going to be transcribed. But if you have the repressor and it's really a co-repressor that comes into play, and usually the co-repressor is the end product, so the product that you're ultimately making at the end. If the co-repressor comes in along with the repressor, they pair up, and now they're able to repress this region here and prevent the transcription from happening. Why does it make sense that the end product, would be the co-repressor? Why is that a logical series of logical thought? Why would it make sense that the co-repressor is also the end product? It's negative feedback, exactly, right? You've made enough of what you need, and so you say, okay, now I'm going to stop more being made, and I'm going to stop it myself, is what it comes down to. All right, so that's what you have going on as far as this regulation is concerned. So these are just two types, two examples that do make the point. So in this case, the repressor is tryptophan, the repressor is active and stops the transcription from ultimately happening. Now, when you think about the order of events, kind of what's happening with transcription, you've got three stages. You've got initiation, elongation, and termination. So the initiation happens so that things get started. That's pretty much the simplest way to put it. You've got various components that are involved. You've got the promoter, you've got transcription factors, you have RNA polymerase, and you have ribonucleotides that are coming into play here. The elongation period, you're going to continue to need RNA polymerase. You're going to need helicase if you are in or a specific helicase, depending on your prokaryotic or eukaryotic transcription, but regardless, you need some kind of helicase. And then, of course, you're going to have more ribonucleotides that are ultimately needed. And then the termination sequence is just a sequence of events that happens that terminates the transcription. So essentially, you're only going to transcribe the piece of DNA that you want to ultimately be transcribing. So here's how this works. What you have is, again, you have your DNA, and similar to DNA replication, a bubble is going to form. The difference here is that instead of replicating the DNA, you are now going to be creating RNA, right? You're transcribing into the RNA. And we're going to go back into the detail on this one here in a second. But really what you care about is pretty much the same kind of order of events. The difference here, though, is that you are not creating two new pieces of DNA. You're creating one single-stranded piece of messenger RNA, right? That's all that's going to ultimately be happening. Now, similar to before, when you were looking at the telomeres, you've got a 5' cap that gets formed. You got a poly A tail that gets formed on the other end, and the reason for these again is bumpers to protect the messenger RNA. But also, what happens in eukaryotes is this phenomenon of splicing. So let me show you what we mean by this. When the transcription is going to occur, the transcription is going to occur for everything, right? The whole kind of from start to finish. So from the prime end to the 3' end, here's your 5' cap that you have. Here is the poly A tail that you have, the other bumper that's on the other end of the chain. These are untranslated regions. This is what's referred to as the 5' untranslated region. This is the 3' untranslated region. What's happening, though, is notice here you've got exon 1, you've got exon 2, and you've got this intron. The problem is that, not the problem, the awesome thing is, our DNA has lots of information stored, and there's lots of kind of overlap as far as the information goes. So in order to pack in all the genetic information that was necessary or that's necessary for the human body or for anything to exist, there's lots of overlap that's built into it. So as a result of that, what happens is you have regions of RNA, of messenger RNA that gets created that you don't actually want or don't need for the expression of the protein that you want to be making. So you've got the exons and the introns. The exons are going to be expressed. The introns are introverted. They're going to stay inside. So what happens is these introns get cut out, and then these two pieces get spliced together. It's kind of like, you know, taking two really old-fashioned, but if you had old movie strips were taken, you know, they cut them and then they would stick them back together. Now we do it digitally, but you're splicing it back together, right? You're taking two pieces, connecting them, getting rid of that middle piece that was ultimately there. And so that new splice piece, that new exon 1 and exon 2 that is all nice and connected, that is going to give you your actual sequence that is going to be translated down the line. All right, so that's what's happening there as far as transcription is concerned. So this is all stuff that happens post transcription. So this is a post transcriptional processing. Now, if we were to go back over here, the important thing to keep in mind is that similar to the way that we're creating our DNA, the same thing occurs as far as the creation of the RNA. So, if you think back to how this is all going to play, you are creating again a 5' to 3', right? So it's the same rules apply as far as the way that we're reading and the way that we're building, except this time we're using RNA polymerase as opposed to using DNA polymerase. So that's the only difference between the two, is that you're using a different type of polymerase to make this all come together. So with that said, let's try question number 4. Now, before we do question number 4, I want you to actually take a look at something more holistically on this one. What is there a lot of on the screen that there wasn't a whole lot of before? What is there a lot of? There's a lot of text. There are a lot of words, right? There's a lot of words. So keep that in the back of your mind as you're going through this question. We'll put a minute on the clock, try it out. I'm going to show you something. Let me show you something that's pretty cool. All right, good luck. All right. So this is a problem, has a lot of text in it, but here's what I want you to take note of. And this is very, very common, very, very common on the MCAT. Notice that the first 3 words in all the answer choices are exactly the same. Transcription from DNA. Transcription from DNA. Now here's my question to you. Where does transcription occur? Where is transcription occurring and only occurring? It's not a trick question. Where does transcription occur? It's in the nucleus, right? It's in the nucleus. DNA, as awesome as it is, and again, I'm personifying DNA, my apologies. This is what I do sometimes. DNA is lazy. It's not going to go anywhere. So transcription from DNA in the cytoplasm cannot be the correct answer. Transcription from DNA in the ribosome cannot be the correct answer. Transcription from DNA in the cytoplasm cannot be the correct answer. So even though there was a lot of text on this and these answer choices, asking you a simple question of where is the mRNA production occurring and the sequence that it's occurring in, you can actually eliminate all 3 answer choices. And this is not coincidental. This is how the MCAT is written. It's not purely a content test. The MCAT is an exam that requires you to know content, but it requires you to prove that you know the content by being able to apply the content and applying the content through a use of critical reasoning skills. And that's exactly what you're doing here. You say, okay, all 4 answer choices start with the words transcription from DNA, so I don't need to read those 4 answer choices over and over again. If you read just the next part, only one of them is factually true, and therefore it must be the correct answer. So for the 76% of you that chose answer choice B, that's correct, nicely done. If you chose D, the problem is it doesn't happen in the cytoplasm, right? It happens in the nucleus, the transcription happens in the nucleus, and the post-transcriptional processing also happens in the nucleus. And then the messenger RNA is sent out in the world. It goes through one of the nuclear pores, it finds itself to a ribosome that's either attached to the endoplasmic reticulum or a ribosome that is free and not bound, and it gets translated. All right, but the correct answer choice on this one is B regardless. You didn't have to read the whole thing. So save yourself the time, be on the lookout for those things. These are written just like the real MCAT. Yeah, a couple of you were asking, is this real? Nadir? Yeah, this is how they are, right? Because here's out of curiosity, just humor me. How many of you read every single answer choice? Just out of curiosity. Like read through all the answer choices until you found the one that you liked. I see a bunch of you are admitting, maybe a couple of you don't want to admit, right? Yeah, it's okay. I mean, to be honest, this is why you're here, is to learn these kinds of things, right? So that's part of the strategy. If you did read it, great, no problem. You only read the second half, that works too, Sabrina. I see a couple of you saying that you read, and some of you read all 4 of them twice. That's fine too. Bottom line though is you can trust the system. You can trust the way the way that this is, and this is how you shave off time. Because here's the thing: for every question that is as quick and simple as this one, there are questions that are long and convoluted, right? So it happens. Yeah. Okay. So Byron, you caught on, right? You're like, wait a minute, this is starting to look familiar. This is starting to look familiar, and then all of a sudden, that's what it comes down to. So, very, very good. Very, very nicely done. Let's take a look at this next one, question number 5. This one is a little trickier. This one is definitely a little trickier. We'll give you a minute. Let's do the same thing. Put your answers in the poll. You've been doing really well so far, so I'm expecting great things from this last one. All right, let's see how we did on this one. It looks like about 55% of you chose answer choice B, 33% of you chose answer choice C. Let's go through this. So this is a fun one because this is, yeah, and I see you're changing your answers, which is totally fine, totally fine. Don't be influenced by what others do. Stick to your guns. We've got a sequence of DNA that is going to be transcribed into RNA. So here's my first question to you, and we didn't talk about this yet. I deliberately didn't talk about this yet. What is it that RNA has that DNA does not have, and vice versa? What is the difference as far as the base pairs are concerned? What letter doesn't show up? Yeah, so we got uracil, we got U in RNA, we don't have thymine. So if we just quickly looked at the answer choices, you get rid of answer choice D right from the get-go, right? So no one should have chosen D because you've got T's all over the place, and you don't want T's. You need D's, right? So here's the next part of all this. You're going to build 5' to 3', but you're going to read 3' to 5'. So we have to do this a little bit backwards. So if we want to go from DNA to RNA, this C here, what is it going to correspond to? What am I going to have as far as my letters are concerned? Actually, let's do it the other way. Let's start with the G over here. I'll do it the other way. So if we start with this G here, what is my first letter going to have to be on the 3' end, on the opposite end, as I'm reading my 5'? What am I going to ultimately be saying? So you're right with the G, it's going to be the C, right? So the G's go with C's, right? The G's go with C's, the A's go with U's, and the T's go with A's, right? That's what we're going to see, and the C's are go with G's. So as we're reading this, we got to go about this a little bit backwards, in that we say, all right, looking at the sequence, we have GAT over here. So what is that going to correspond to? It's going to correspond to CAU over here, right? Or you could have done it the other way around. You could have said, I'm going to read the C, so I'm going to end up with the G. I'm going to read the T, I'm going to end up with an A. I'm going to read the G, I'm going to end up with a C. So the correct answer on this one has to be answer choice B. Kind of looks like guacamole, but it isn't. All right? So that's the one that you're going to want to choose, and that's the one that's going to get you the points. So B is the correct answer. B is definitely the correct answer. So with that said, this is the beginnings of molecular genetics. That is what you see on the test, or the types of stuff that you're going to see on the test. There's obviously more. There's definitely translation that occurs next, which we're not going to go into here tonight. But bottom line is, as you're studying this stuff, it's really, really important, for you to make sure that you are putting all this stuff together and making sure that you're kind of taking this from the perspective of not just the content that's in front of you, but what's the detective work that you're going to need to do. In fact, there's a lot of detective work that ends up having to happen as far as this is concerned. So something to keep in mind, something to take into account, as it were. With that said, I'm going to switch. I got a question mark in my mind, actually. I'm going to switch us over to Allie, but Allie, there's some lightning, or there's some storms that are coming wherever Allie's in Florida, if I remember correctly. So I don't know if Allie's going to be able to come back on cam. Allie will let me know here in a second. But I want to take this opportunity, at least from this part here, to give you all something. We reserve these. I think Ashwarya and Allison and Candace will all back me up on this. Mackenzie will back me up on this. Sharon will back me up on this. We don't give out a lot of these, right, guys? These are pretty rare, what I'm about to do here. I'm going to give you guys a star. I'm going to give you guys a green star. So there's a nice green star for your work. You guys did awesome. Hopefully what you saw here, gives you a good sense of kind of where it is that you need to start working on, and the things that you need to start working on, and where it is that you need to go. But let's continue the conversation. Let's figure out. We've looked at some science. Hopefully you had some fun with this. Yes, I think you did. I'm seeing a bunch of thank yous, so you're all very, very welcome. A couple of you were asking, well, when's the translation lesson? We're not actually doing that as part of Free Prep Week. We're only doing transcription, the first part. So it's not part of Free Prep Week. There is a translation lesson that actually I'm teaching, I think, relatively soon on the MCAT channel. So it is there. But let's talk about the do's and don'ts. If you got questions about this stuff, let us know. Specifically, and let us know what those might be. Let's talk about just studying for the MCAT. I know a bunch of you are taking the test this week. If you're taking the test this week, if you're taking the test Friday or Saturday, don't study tomorrow. Like, take the night off if you're taking the test on Friday. Take Friday off if you're taking the test on Saturday. If you're studying next week, never study the day before the exam. That's like the worst thing that you can do. Cut the pie. Yeah, good old pure as gold, right? Pure as gold. That's how you remember. The purines are A and G. Cut the pie. That's how you remember the pyrimidines. All those fun mnemonics, right? All those fun mnemonics that comes into play for sure. But let's talk about the do's and don'ts. I'm going to just switch us back over to our other room here. So give me a quick second to load these slides up and also make sure that my camera is working and making sure that you all can see what it is that you need to see. So first and foremost, I'll say this much. You don't want to study from textbooks. Has anyone done that or started doing that at any point when you're studying for this exam? Was that a tactic? You were like, I'm just going to open up my old textbooks. I'm going to go to the library, get some old textbooks and see what I would do. Carol, I totally agree with you. Studying the last minute is the worst thing. It's just not great, right? It just feels bad. So yeah, it's, the thing about the textbooks, the thing about college textbooks specifically, we'll get into the Kaplan books in a second here. The thing about college textbooks is that they have so much more information than you will ever actually be tested on. Even if you look at your notes from school. So Autumn, I hear you. You look at your notes, you say, okay, let me go back and see what my professors were saying. They're giving you more information in there than you're going to be responsible for the MCAT. So think of your, my OChem textbook. I still have it, 1,500 pages, awesome textbook, far more information than is tested on the MCAT, right? Bottom line. So studying from textbooks, definitely not a great idea, namely because you're going to overstudy and you're going to burn yourself out. As you're studying for the MCAT, it comes down to this. So here's what I tell every single student that I talk to. So you're going to hear it. Earlier on in your preparation, you're going to be doing a lot of content review. As you move away from the beginning, that content review needs to diminish so that you can increase your practice. So the best way to retain information, the best way to retain knowledge is to practice. The application of the material is the best way for you to actually learn it. I get the question a lot of, how should I memorize formulas? My answer is simple. Do lots of problems, because the more problems you do, the more second nature the formulas are going to be, right? For those of you that drive, or if you ride a bicycle, you don't think about what you should be doing. It's all muscle memory, right? You've learned this and it's become innate and it's become something that you do. Same thing is true with the MCAT and with the content. The more drills you do, the more practice you get, the better you're going to become at it. Yes, you still are going to go back and review things. Yes, you're going to still need to go and, I have some students who are taking the test this Friday, and yes, they're going to be looking up things as far as content goes. But the bottom line is, you're not going to want to be spending all of your time doing that content. You're going to want to be investing a lot of time actually getting practicing. So part of that, of course, is setting up the right study plan. So the cool thing that that we do as part of our program, and something that I hope that you will take advantage of, is we actually help you build out a study plan. And it's done very, very scientifically. It's actually kind of nerdy the way we do it. We give you a diagnostic MCAT, and we give you a separate assessment. It's a science assessment that has, the questions aren't in MCAT format. It's just straight, do you know the material, do you not know the material? That gets fed into this really awesome algorithm that actually generates a customized study plan for what your individual needs are. So I'll tell you my story. I don't oftentimes share the story, but I'll share the story with you guys tonight. When I was in undergrad, I didn't care for physics. I had AP'd out of it, so I didn't need to worry about taking physics. MCAT rolls around, and I hadn't taken any physics in like 3 or 4 years. And I was a biochemistry major, so I had taken a ton of chemistry. So I did just fine on the Chem Phys section, but it was all because of my chemistry. It wasn't because of anything in my physics. So when I was studying for the MCAT, and this was my diagnostic, I'm saying, I did, just fine, not well enough, or not as strong as I eventually did. But when I was studying, the place that I put my focus on, it was simple. It was going to be physics, because that was the area that I hadn't really touched in a long time. It's not as straightforward, right, for most folks. I mean, there's different areas, different strengths and weaknesses, and all that stuff. So what our diagnostic assessments do is they actually tell you, like, here's the stuff that you need to be working on. Here are the exercises that you need to go to in order to make sure that you're doing the right thing. So kind of number one is make sure that you are constantly assessing yourself so that you make sure that you are working on the things that are right for you. The last thing I want anyone here, any MCAT student doing is just studying stuff for the sake of studying stuff. You need to be doing the work you need to be doing in order to be improving your scores. So there's a little mantra that we have around here. Forgive me if you've heard it before. It's to be efficiently lazy, right? So if you do the work that you need to do in order to be improving your scores. So getting lots of drills, getting lots of realistic practice is a huge part of it. The benefit of taking full-length exams, I can't emphasize how important full-length exams are. But I will say this much. You don't want to just take full-length exam after full-length exam after full-length exam. Why is that not a good idea as far as your preparation? To just take one full-length exam after another and do nothing in between. Does that logically make sense as far as a good idea? Yeah, it would be total burnout, absolutely. And you wouldn't actually be learning anything from taking the full-length exam. The act of taking the full-length exam is to give you simulation for a 7.5-hour exam that's going to be really hard. The point of taking full-length exams is to go through the full-length exam, review the materials that you have gotten wrong, right? Yeah, you're absolutely right. You guys I'm loving this. So watch the stuff to see what it is that you need to be doing. What is it that you need to not be doing? And then from there, take it into account and say, all right, here's where I'm going to ultimately be putting my focus. And then you do that studying and take another full-length exam. So generally you'll run into a cadence of about one full-length exam per week. So approximately speaking, it's about one full-length exam per week is probably what you're going to want to be doing as far as the MCAT is concerned. So make sure that you get yourself access to at least 6 full-length exams. It's a huge part of your preparation. We have I think we're at 15 full-length exams now in our course. So you've got all the ones from the AMC plus 11 that are from Kaplan. So you got more than enough full-length exams as part of our course, and it's a nice little feature that you have as you're moving through it. And then at the same time, of course, you want your sources of expertise. So those are your teachers. Those are, like in our course, you've got the MCAT channel. You've got, if you sign up for one of our plus programs, you'll be paired with a med school coach, med school mentor. These are individuals who are not only MCAT teachers, they are MCAT teachers who have been accepted into medical school and been through this process. They're not going to, walk you through. It's not like a tutor that will walk you through subject material. They're going to walk you through and help you set up a customized, like having a personal trainer, customized plan based on what your needs are, and you build a relationship one-on-one with them, similar to how you'll build a relationship with your instructors that are in your classes. So these are this is kind of the overarching ways to be thinking about what it is that you need to be working on. If you are, and I saw somebody who said that they just signed up for a course, so that's kind of awesome. I forget who it was now. Maybe we can find their name. So congratulations. We'll see you on the MCAT channel. The way to think about it was Buck. Thank you, Ashwarya. I remember I was looking and I was trying to remember who it was, but I couldn't find it in the chat. So the way that our program is set up is we lots of different options available as far as how it is that you want to take your course and all that stuff. So if you've got questions, let us know what they are. Jamie just asked a question. I'm going to answer this one. What are the pros and cons of doing an online class versus an in-person class when you're prepping for the MCAT? So here's our take on it. regardless of which modality you decide on. So Jamie and everybody else is kind of trying to figure out what it is we need to be doing, to improve our MCAT score. Whatever modality it is you decide to choose, that's probably going to be the one that works well for you, right? So if you're the type of person who wants to go to a physical location, in person is a good option for you. The advantage you have with live online, though, and I'm not biased when I say this. I have taught both in-person classes and live online classes, and I've done tutoring, right? So I'm speaking from experience, but also speaking from what our students say. The advantage of a live online class over an in-person class, number one, obviously the convenience, because you don't have to go anywhere, right? You're just like, I'm gonna do this from my house. I'm gonna be in my, pajamas, and that's totally fine. So the convenience is a nice factor of it. The bigger part, though, is that our live online classes draw faculty from all of Kaplan throughout the entire organization. So you are not limited to the instructor who is local to your area. So that's number one as far as the pro of a live online class. The second part, though, is that with the live online class, not only do you have the instructor who's on camera, you actually have a team of instructors. So tonight you have Sharon, Mackenzie, Candace, Allison, and Ashwarya, because we got a big group here. When you are in a live online class, you don't wait to ask your question. So if you've been through this, right, you're in a class, you raise your hand, right, you wait to be called on, you ask your question, the instructor who's at the front of the room is going to answer your question, and then somebody else asks the question, right? That's a traditional in-person class. With a live online class, you have a dedicated team of folks who are not on camera. They sometimes are on camera, right? I TA sometimes, and others teach. There's different. We all do different things, but we're all teaching just different modalities. So in a live online class, you ask your questions and you get them answered. So we've actually found that live online courses are more interactive than in-person courses because of the fact that you are instantaneously getting your questions answered. You're kind of able to work at more of your own pace, even though you're in a group learning environment. Aside from that, everything else is the same. You get the same books, you get the same online materials, all the questions, you got the 10,000 questions. This says 14 full-length exams. It's actually 15. We just haven't updated the slide yet. My apologies. We'll get that fixed. You get the same higher score guarantee. You get the MCAT channel. Nice thing about the MCAT channel is even if you're taking an in-person class, you've got access to the MCAT channel. So everybody has access to the MCAT channel as far as our comprehensive courses, except for students who are taking self-paced. Self-paced is a kind of on your own program. There's no MCAT channel attached to it. Self-paced Plus does come with the MCAT channel. So hopefully, Jamie, that answered your question about that specifically, but that's the bit there. The one thing I will say as far as the online course con is that you do have to make sure that it's really easy when you're taking an online course to, have open your chat and maybe your Facebook account and you're texting your friends and all that good stuff. So you have to just make sure that you kind of close yourself off for that. But that's actually a really easy fix. That's not a very difficult fix at all. What other questions do you guys have? Like, what are the other things that are going on in your mind? You're very welcome, Jamie. Absolutely. There's a lag between your chatting and me seeing it, so my apologies for not seeing that. Thank you. Just yet, but until then. What other questions do you guys have? I'm just curious to see like what's on your mind as far as what to do for this thing. Like what's keeping you up at night as far as the MCAT. Let's start there. I'm just curious to know what you're all worried about. Lordsy had a good question. Is the online package beneficial for tactile read-write learner? Absolutely. Yeah, absolutely. It's not. The online package is, the online course, I should say. It's not, it's not like online and all of a sudden everything changes. We still send you books. It's kind of weird. You're like, I bought an online course and now you're sending me books. So yeah, we still send you books. So, and if you were in an in-person class, you would get books. Everything on that end remains the same. So, Is 1 month, Patel asks, is 1 month enough time to start studying plus practice exams? Here's the thing. Generally speaking, we recommend between 300 and 350 structured hours of preparation. That includes class time, taking practice tests, and then all the studying that you're going to do on your own with the right kind of level of detail and the right type of study planning. In a month, that's going to be very, very hard to achieve. So unless you've studied already for the exam, definitely something to keep in mind. I wouldn't rush it. A month is pretty tight, but I've seen students do it. So I'm not saying that it can't happen. I've seen students do it, and that's the part there for sure. Yeah, let's see what else we have here. I've seen some good questions coming in. There's a question from Katie. The question was if there's instruction regarding biochem and psych in the non-plus courses, and the short answer is yes. So this is a little bit misleading. Our course, traditional course, non-plus course, I should say, covers all the biochemistry, all the psychology, covers all the subjects that are on the MCAT. If you take the plus program, we give you something called Foundations of Biochem, Psych, and Soc. The Foundations course is designed for somebody who has never taken these courses in school before. So the expectation when you take an MCAT course with us is that you've taken these courses once and you've seen them, and you're reviewing the material and you're relearning it the way that you need to learn it for the MCAT. There is a population of students out there that haven't taken biochem, psych, or sociology because most medical schools still aren't requiring that as a course requirement. This is the discrepancy. We didn't talk about this at the beginning, but this is the discrepancy between the pre-med requirements and the MCAT requirements. MCAT requires biochemistry, psych, and soc. The pre-med course requirements that you need for your application don't require biochemistry, psychology, and sociology. So the plus program gives you this foundation course, which essentially just teaches you the biochem, teaches you the psych, teaches you sociology from the ground up, like assuming zero prior knowledge. Imagine it's like day one. In fact, we've had a lot of students who've taken the Foundations program before going and taking biochemistry in school, so they get a better grade at the end of the day. So yes, the course is going to cover everything. It's it's a fully way to do so. I saw another question, and I can't remember. Shwarya, maybe you can remind me who asked it. As far as there's so many resources. Where do you start? And the place you start is you start at the beginning. You take the diagnostic, you take the science assessment, and then that will govern to you what it is that you need to be working on. So really the best place to start usually is by attending MCAT channel sessions prior to your course starting, and then using the adaptive QBank. So using the adaptive QBank allows you, the way that our new QBank works is actually new for 2018. The way the QBank now works, the QBank represents a question bank. So there are about 2,600 or so questions in the question bank. What it does though is you pick the topics, you pick how many questions you want to be doing, and it gives you kind of a middle level question. And if you get that one right, it gives you a little harder question, but if you get it wrong, it gives you an easier question. So what it does, it tries to find your zone of proximal performance. So you're always being challenged, and therefore you're always learning. So you're never bored with the material that you're ultimately looking at. I want to see if I have a slide here for it, but this is a slide for the MCAT channel. That's Eli. Eli's actually, he just finished teaching on the MCAT channel. I was teaching at 7, and then Eli taught at 8 and 9 earlier tonight on the MCAT channel. So, but you guys saw the MCAT channel tonight. We just did a lesson from the MCAT channel, so nothing new to report there. But the QBank, pretty awesome technology that lets you find those pieces and take it from there. Ben, you're taking the MCAT in 2019. When do you start studying? Great question. Depending on when you're taking in 2019. So if you're taking in January 2019, I'd actually say start studying this summer. Kind of get the material going, and then start in earnest in the fall. That's what a lot of students do. Is they'll sign up, they'll get the books going, and then they'll start in the fall. If you're taking the test January, if you're taking, generally speaking, you want to give yourself 3 to 5 months. What it comes down to. So, Jessica, you like the QBank? I'm glad you like the QBank. Yeah, I'm happy. We just actually that's a new thing. I mean, we have the QBank as part of the comprehensive course at the beginning of the year. We just added it. It's something you can actually get just the QBank on its own. So if anybody just wants QBank, if you go to our website, you can see there's different subscriptions, so you can get it for different periods of time. We also have a couple of practice packs, which just give you 3 full-length exams, or you can get one that just gives you science review videos. So yeah, thank you. There's the link for QBank, and then maybe Ashwarya can put the link in for the prep packs as well. There we go. See, Ashwarya's got it. So it's pretty awesome in that regards. Michaela, we're asking about how many 1-hour lectures are there on the MCAT channel. Right now, there are about 100. We're in the process of doing some updates and adding a few and combining a few and, kind of shuffling them around, if you will. So there are about 100. I want to say there are like 91 that are active today, which is a lot. We don't expect you to go to all 91 of them at all. You can access the lessons anytime you want. So they are available in an archive, but then they're also offered live every day. So I shouldn't say every day except for Fridays. Kaplan has an old tradition of not teaching on Fridays because in the old days, there were most of the standardized tests were on Saturdays. So we wouldn't teach on Fridays because you don't want to teach or you don't want to take class before the test. But so 6 days a week, you've got MCAT channel live, and there's usually 3 or 4 lessons that are being taught on any given day. So, and they just kind of rotate, right? So they just kind of rotate as it were. So you can access them anytime, and they are on the highest yield topics. So you will be, the different, there's so many different resources in our program that we have selected the right type of learning modality for each of the topics and each of the, whether it's high yield or low yield or whether it's hard or it's easy. Some stuff, it works just fine in a book. Some stuff needs videos and a book. So some stuff needs live instruction. So you got the MCAT channel. Some stuff needs to be in the core lesson. So you have that in the core 36 hours. Altogether, you have 125 plus hours of live instruction available as part of your program. So that's the bit. So that's what we got there. Let's see. What advice? Oh, that one disappeared. Test anxiety. Test anxiety is actually, it's a big problem. We actually have a bunch of things that we do in our course to help you with test anxiety. I'll give you this much. In the time that we have here, the best thing to do to overcome test anxiety is to rest. So that's breathe, rest, and don't get fixated on the test. Get fixated on getting more questions right. That's the bit there. There was a question about. I don't see where it went. It must have disappeared. There was a question that was a good one, which now has disappeared. So I'll try to remember what it said because it was a good one. It had to do with the course, but now I can't find it. Maybe Shwari, you grabbed it or got it answered. Yeah, so lots and lots of stuff to look at. Boot camp. Tomorrow night's boot camp. So if you want to close out the week, come to boot camp. Boot camp is a little different from tonight. Boot camp is more question driven as opposed to any content. So it's a special event that we do. It will be the same as previous boot camps. So if you've been to a Kaplan MCAT boot camp, it's going to be the same, and that's what it is. Julie had a question: if you sign up for online classes that will start later this summer, will you be sent the books and get access? And the short answer is yes. So as soon as you enroll, we send your books. We send your shipping order, and off your books go. Right. So that's the way it works. So as soon as you sign up, Julia, your books will be sent to you. You'll get access to the channel. You can actually sign up tonight. And you can start going to the MCAT channel tomorrow night. You'll get the QBank, all that good stuff. It all activates as soon as you get enrolled. And then, whenever it is that your actual core class sessions start, that's when your core class sessions start. So, I had a student in channel last night who I taught a lot of channel this week, but I had a student in channel last night who, their core course isn't starting until June. but she just signed up and she wanted to start coming to channel because she's waiting for the core course to start, or the core part of her course. And so that's what it comes down to. So. Jamie, that was Jamie. It was your question. Yes, there it was. The course plan question. Thank you. Yes, the course plan does take into account when you're planning to take the actual MCAT. So the way that the study plan is set up is, you expire during the month of your exam, or the month after your exam, basically. So yeah, so it's spaced out accordingly. The calendaring you're still going to need to do, but that's because you've got to build your own calendar. We can't build a calendar for you. But yes, the way that it's built out is that we give you the recommendations for each of the different topics, each of the different areas, and then it's, that's your responsibility then to say, okay, how am I going to space this out when I'm actually taking the test? So it sounds like if you're thinking 8 months, you're probably taking the test next year. That's what I'm guessing, but I might be wrong. Let us know, and that can we can help you figure that part out for sure. Cool stuff. There's one last thing I wanted to share with all of you. Two things, actually. One, if you want to meet Aishwarya or Allison in person, you can, because they're going to be teaching at our summer intensive programs this summer. We always have classes in the summer, just our regular classes, but then we have a special program. It's called our intensive program. And the intensive programs are offered both in person. We have it actually starting in Atlanta this week. We've got Boulder coming up in a couple weeks, right? And then we've got San Diego a couple weeks after that. These are 6-week boot camp. Pretty much you do nothing but MCAT the whole time. And I know Aishwarya and Allison are teaching there. I'm not sure if anyone else is this summer. So you can meet them there. We've also got the same summer intensive programs available as part of Live Online. So this is different from taking a course live online. This is a different program. It's a more structured program, but it's an awesome program. So if you're thinking of like the only thing I want to do this summer is study for the MCAT, then summer intensive might be an option for you. And if you go to San Diego or to Boulder, you can meet Allison or Aishwarya. So that's some opportunity for you there. The other thing I wanted to just remind everybody of, two things actually. One, there is, if you go to our website, you can actually see that there's some savings. I saw some questions that came in earlier about, affordability and all that stuff. So, bottom line, if you go to our website, there's actually a sale that ends tomorrow. Tomorrow's Thursday, right? Yeah. So there's a sale that ends tomorrow. So you can save a little bit of money. I think it's $200 off of our in-person and live online courses and self-paced, and then $300 off if you wanted to do a plus. So there's opportunity to save a little bit of money there. We do have installments that are available. So if you're thinking of doing a course, but you don't want to, put everything up front, you can do installment billing. So that is available as well. You can break it up into 3 parts. But we also have tuition assistance. So as Ali mentioned at the beginning, just a reminder, if you are thinking of taking a course with us and you're worried about the tuition costs, there's that as well. For the whole month of May, we're actually also including, we relaunched, I should say, some of our admissions consulting offerings. So we actually have an admissions help package that's also included, and that's the whole month of May for our MCAT programs. But the dollars off savings ends tomorrow. So if you're thinking about taking a course with us, now's a good time. I mean, I'm not, again, like no pressure, but just something to be thinking about, because especially if you're planning on taking the test this summer, so that you can apply this year, now is pretty much the time to be getting started on that. As far as Julia, as if you miss a class, is it recorded anywhere? Yes. So we have all the lessons are recorded and available to you 24 hours a day, 7 days a week. But you can always, if you miss a class, you can always make it up. So you just we walk you through finding a makeup opportunity, and you can do that as well. I didn't show you this. This was the last thing I want to show you guys. This is the smart reporting system. So remember I said we make recommendations for you. This is part of what it looks like. So it's you can't really see it here too well, but it's a very detailed thing. It's like you need to work on concentrations, and then it actually, on a separate page that we don't have here, shows you what the exercises are that you need to work on, right? So it actually walks you through all the bits along the way. We have courses available and live online, in person, self-paced tutoring. If you want more information, you wanted to talk to one of us, you can either give us a call when you're at Cap Test, or you can email Ali. There's the links at the top of the page there. You can email us at consultateacher@kaplan.com. That's actually all of us. Ali is mostly looking over it this week. But if you want to set up a time to figure out what your right prep options are, if you want to kind of talk through and figure it out, click on the link that says kaplanteacherconsult.youcanbookme, or youcanbook.me. Click on that and you can actually schedule an appointment, and somebody will reach out to you at the time that you want them to reach out to you. And these are teachers who will reach out to you. So you can, I might reach out to you. Who knows? I might be the one who calls you. But we can take it there from there and kind of figure out what the right thing is for you, and tell you all about like the higher score guarantee and how that works, which is we guarantee your score will improve, we'll give you your money back, or you can continue studying with us. So it's a matter of you figuring out what's right for you. And I see a couple of you are already, Buck, I know you signed up, and it looks like a couple of you are already thinking ahead and figuring out what is it that you need to be doing to get yourself ready. So that's all available to you, and that's what we're here for. But with that said, it is 10:30, so I'm very sad to say that our time together for this evening has come to a close. And so thank you all for being here tonight. Big thank you to Ashwarya and Allison and Candace, Greg and Mackenzie and Sharon and Eric. Eric and Greg were manning things behind the booth to make sure that everything worked as far as the technology. So big thank you to the two of you. Big thank you to my colleagues, my teacher colleagues. A big thank you to each and every one of you for joining us here tonight. Hopefully you learned something. You learned something about yourself. You had some fun. And now it's a matter of figuring out what's right for you and figuring out what is it that you need to be doing for the MCAT, and for really, think about it. Achieving the goal, the dream that you've been looking to achieve, which is wearing a white coat. That's what this is all about. So we'll see you back here maybe tomorrow night for boot camp. If we don't, I hope our paths will cross again. Thank you all again for joining us tonight. And again, big thank you to the team. And sorry Ali got sidelined by some storm. So hopefully Ali's okay. I'm sure she is. But we'll put it all together tomorrow at the boot camp for sure. Thanks all. Have a great night.